Biochemistry 401 Lecture 29. Today we're going to talk about cholesterol. We're going to talk about the function of cholesterol first, and then the synthesis, the regulation, and some cholesterol derivatives. And this will include the function and the synthesis of these derivatives. So let's get started. Cholesterol is a steroid consisting of 4 rings and a hydrophobic tail. It is a very planar molecule and hydrophobic. It functions as a membrane lipid to modulate membrane fluidity, and it's also found in lipid rafts. It is a precursor for synthesis of steroid hormones, of bile salts, and also of vitamin D. Now, cholesterol can be transported in lipoproteins and can be stored in the cell, but to do so, we're going to hide the polar hydroxyl because that is the only polar group in the whole molecule, and we're going to hide that by adding a fatty acid through esterification. We're going to make an ester linkage between the fatty acid and cholesterol to make a cholesterol ester. This is going to increase the hydrophobicity of cholesterol and allow it to pack even tighter into a smaller area for storage in the cell or for transport. In membranes, the polar hydroxyl group is important. It's oriented towards the aqueous environment and interacts with polar head groups of membrane lipids and water molecules. There are 2 sources of cholesterol, dietary cholesterol and endogenous cholesterol. Dietary cholesterol is ingested in the foods that we eat. It's packaged into transport micelles, and this happens in the intestines. These micelles are called chylomicrons. Endogenous cholesterol, on the other hand, is synthesized de novo. It is packaged up in the liver into transport units called very low-density lipoprotein particles that include both lipid and protein, and we'll talk about both of these later on in more detail. Here we see a brief overview of the steps involved in cholesterol synthesis. There are 5 important steps. Step 1 is a condensation of 3 activated acetyl units to form the 6-carbon molecule HMG-CoA. Step number 2 is the reduction of HMG-CoA to form mevalonate. Step number 3 is decarboxylation to the 5-carbon activated isoprenyl unit, isopentenyl pyrophosphate. Step 4 is the condensation of 6 activated isoprenes, both isopentenyl pyrophosphate and its isomer, dimethylallyl pyrophosphate, to eventually form the 30-carbon unit squalene. This happens first through the formation of geranyl pyrophosphate. 5 plus 5 equals 10, and geranyl pyrophosphate has 10 carbons. We're then going to add another 5 carbons to geranyl pyrophosphate to yield farnesyl pyrophosphate. Farnesyl pyrophosphate contains 15 carbons. Then comes the condensation of 2 molecules of farnesyl pyrophosphate to form squalene. 15 plus 15 equals 30. Finally, step number 5 is the cyclization of squalene to form linosterol, and then modification in several steps to form the mature cholesterol. We're going to look at these one by one in more detail. The first reaction that occurs in cholesterol synthesis is a condensation reaction between the 4-carbon molecule acetoacetyl-CoA and the 2-carbon molecule acetyl-CoA. This results in the production of 3-hydroxy-3-methylglutaryl-CoA, HMG-CoA, and a release of coenzyme A. Now, we've seen HMG-CoA before in the mitochondrion. It's also an intermediate in ketone body synthesis. And so here we see 2 isozymes of HMG-CoA synthase, one in the cytoplasm that functions in the synthesis of cholesterol, and one in the mitochondrion that's involved in ketone body synthesis. Now, in the cytosol, 3-hydroxy-3-methylglutaryl-CoA is an intermediate in cholesterol synthesis that is going to be reduced in a series of reactions to yield mevalonate. And the electron carrier in this case is NADPH. And so what we're left with are 2 molecules of NADP+, coenzyme A, and the 6-carbon intermediate mevalonate. Now in the mitochondrion, as we said, 3-hydroxy-3-methylglutaryl-CoA is an intermediate in the synthesis of ketone bodies, and it's used to make acetyl-CoA and acetoacetate. You'll recall that acetoacetate is actually a ketone body. And so let's go back to the synthesis of mevalonate in cholesterol synthesis in the cytosol. In going from 3-hydroxy-3-methylgluteryl-CoA to mevalonate, there are two reduction reactions, one to produce an aldehyde and the second to produce an alcohol. The enzyme that carries out these reductions is HMG-CoA reductase, and this name makes sense. This is actually an integral membrane protein that carries out a cytosolic reaction, but the protein itself is anchored to the outside face of the endoplasmic reticulum. HMG-CoA reductase catalyzes the rate-limiting step in cholesterol synthesis. It's the slowest step in the pathway, and therefore it's highly regulated. Now, the next step that we're going to see is going to use 3 molecules of ATP, which is really expensive. And so it's a good idea to make sure that you really want to make mevalonate. Here we see a figure of one monomer of the tetrameric enzyme HMG-CoA reductase. In eukaryotes, this is an integral membrane protein that's bound to the outer face of the endoplasmic reticulum. This represents one of the most highly regulated enzymes in the human body. And so from the 6-carbon intermediate mevalonate, we're going to produce the 5-carbon activated isoprene unit, 3-isopentenyl pyrophosphate. To do this, we're going to use 3 molecules of ATP. The final step is going to release carbon dioxide to go from a 6-carbon unit to a 5-carbon unit. 1 molecule of inorganic phosphate is also going to be released. You do not need to know all of the intermediates between mevalonate and 3-isopentenyl pyrophosphate. You do need to know the starting reactants, which are the 6-carbon mevalonate and 3 molecules of ATP. You also need to know the final products, which are the 5-carbon 3-isopentenyl pyrophosphate, carbon dioxide, and 1 molecule of inorganic phosphate. Now that we have 3-isopentenyl pyrophosphate, what can we do with this? This is a very important intermediate, it turns out. These are some of the derivatives that can be made from 3-isopentenyl pyrophosphate. They include cholesterol, of course, but also steroid hormones, bile salts, and vitamins D, A, E, K, which you should recognize as being all of the lipid-soluble vitamins. Also, the quinone electron carriers, ubiquinone, ubiquinol, and dolichols. You are only responsible for those that are bolded and are not responsible for knowing the derivatives that are found in plants. Isopentenyl pyrophosphate exists in equilibrium with its isomer, dimethylallyl pyrophosphate, and the difference between these two intermediates is the position of the double bond. Both of these isomers are used in the next step of cholesterol synthesis. The next series of reactions are driven forward by pyrophosphate hydrolysis, and the first reactions occur through addition of isopentenyl pyrophosphate. Reaction number 1 condenses dimethylallyl pyrophosphate with isopentenyl pyrophosphate, and there is a loss of pyrophosphate. This forms the 10-carbon intermediate geranyl pyrophosphate. Then there's another 5-carbon addition through condensation of geranyl pyrophosphate with isopentenyl pyrophosphate to yield farnesyl pyrophosphate, and this is a 15-carbon intermediate. And finally, we condense 2 molecules of farnesyl pyrophosphate to make the 30-carbon squalene. Now, the synthesis of squalene is catalyzed by squalene synthase, and this enzyme catalyzes the first committed step of cholesterol synthesis. And if we think of this, this makes sense. Both geranyl pyrophosphate and farnesyl pyrophosphate can be used as lipid anchors for proteins, and we've just seen the derivatives of isopentenyl pyrophosphate. And so, with the synthesis of squalene, we have the first committed intermediate in cholesterol synthesis. The last series of reactions involve first a cyclization of squalene to form linosterol, and this cyclization is catalyzed by the enzyme oxidosqualene cyclase. This forms a reactive epoxide that spontaneously rearranges to cyclize squalene and form lanosterol. And then from lanosterol to cholesterol, there's a difference in saturation and the loss of 3 carbons. So what you need to know from this is that oxidosqualene cyclase cyclizes squalene to lanosterol, and lanosterol is then changed to cholesterol. And that's it. So now let's talk about the regulation of cholesterol synthesis. We're going to talk specifically about the regulation of HMG-CoA reductase, which catalyzes the rate-limiting step, but the regulation of squalene synthase, which catalyzes the first committed step, is also regulated in similar manner. HMG-CoA reductase is regulated at the level of transcription through the action of transcriptional regulators that bind to sterol response elements. Sterol response elements are stretches of DNA that lie just upstream of some genes that are activated in response to low levels of sterols. And there are several genes that have SREs. HMG-CoA reductase is one of them. When the transcriptional regulator binds to the SRE, coactivators will then bind and will upregulate the transcription of the HMG-CoA reductase gene. Regulators that bind to the SRE are activated and sent to the nucleus in response to low levels of cholesterol in the cell. When there isn't very much cholesterol around, these transcriptional regulators are activated and sent to the nucleus. They then bind to the SRE and upregulate gene transcription. Now, at the level of translation, there is negative feedback from downstream metabolites of cholesterol synthesis that bind to the HMG-CoA reductase mRNA and prevent its transcription. In post-translational regulation, we have 2 main methods. First of all, there's something called a sterol sensor that's found in the HMG-CoA reductase protein. This senses the concentration of cholesterol. When the concentration of cholesterol becomes too high, it causes a conformational change in the HMG-CoA reductase protein. This makes the protein more sensitive to proteolysis, and so the protein is actually inactivated this way. And finally, phosphorylation. This occurs through the activation of an AMP-activated kinase. This is the same kinase that we saw previously in the regulation of fatty acid synthesis, and it becomes activated when energy levels in the cell are low, as evidenced by an increase in AMP relative to ATP. And so when AMP concentrations in the cell increase, the kinase is activated. It phosphorylates HMG-CoA reductase, and it inhibits its activity. This makes sense. Remember, there are 3 phosphorylations in the next step in the process of going from mevalonate to 3-isopentenyl pyrophosphate. And so now I have a question for you. This again is a figure that shows the HMG-CoA reductase monomer, and we said that it has a sterol sensor that senses how much cholesterol is actually in the cell. Which portion of this protein would you expect to be sensitive to levels of cholesterol in the cell? Would it be the exterior that's shown in ribbon diagram? Would it be the transmembrane helices that are shown in blue? Or would it be the loops that extend to the interior of the endoplasmic reticulum? Which of these 3 regions do you think is sensitive to cholesterol concentration? That's right. The transmembrane region is sensitive. These 5 helices that are shown in dark blue represents the sterol sensor portion of this enzyme. Very good if you got this. One of the important jobs of cholesterol is its use in the production of bile salts. Bile salts are a component in bile. Bile is a watery substance that's secreted by the liver and contains organic and inorganic molecules. The organic compounds are phosphatidylcholine and bile salts. Bile is secreted by the liver, it's stored in the gallbladder until it's needed, and when you have a meal, it's released into the small intestine. Bile functions to emulsify dietary lipids. So, what we want to do is we want to make cholesterol more soluble so that it's a good detergent because we want to emulsify lipids. Cholesterol is normally very hydrophobic except for that one hydroxyl. So, since hydroxyls are a really good idea, we're going to add more hydroxyls to cholesterol. We're also going to shorten that hydrophobic tail. And then we're going to oxidize carbon number 24 so that we can add coenzyme A. We're going to do this because we're getting ready for synthesis. The first thing we're going to do is we're going to add hydrophilic hydroxyl groups. Now if you'll notice, these are all pointing away from you, so that we're going to have a hydrophilic face that's pointing away from you and a more hydrophobic face that's pointing towards you. The next thing we're going to do is we're going to shorten the hydrophobic tail and then add coenzyme A to make a thioester. This is known as cholyl-CoA. We can then add amino acids at carbon 24 to make this tail more hydrophilic. We can either add glycine or we can add another type of amino acid that's not used in protein synthesis, but nevertheless is an amino acid that has a sulfate as one of its functional groups. This is called taurine. When we add taurine, we end up with taurocholate, and when we add glycine, we end up with glycocholate. Each of these tails help to make the bile salt more hydrophilic. Now another thing that we can do with cholesterol is we can use it to make vitamin D3. Another name for this is cholecalciferol. The site of vitamin D synthesis is the skin, and what we're going to do is we're going to use the energy from the sun to actually cleave a bond to make pre-vitamin D. This is then going to rearrange to form vitamin D. We need vitamin D for bone growth and for remodeling because this vitamin helps to regulate calcium and phosphorus levels in the blood. It promotes the absorption of calcium and phosphorus in the intestines. And it also promotes the reabsorption of calcium in the kidneys. Another thing that cholesterol is important for is the synthesis of steroid hormones. These include progesterone, which can then be used to make cortisol, aldosterone, testosterone, and estradiol. Progesterone is synthesized in ovarian follicle cells and during pregnancy in the placenta. It is also synthesized in males in the testes, and in both sexes, it's also synthesized in many non-reproductive tissues as well, including the adrenal glands. It functions as a precursor for all other steroid hormones. And in females, it's important in reproduction for the preparation for and the maintenance of pregnancy. These corticosteroids are synthesized in the adrenal cortex. The adrenal cortex is a portion of the adrenal glands that synthesize corticosteroids. We can think of the adrenal cortex as being the rind of the adrenal gland, and the adrenal medulla is the center of the gland. You'll recall that the adrenal medulla is important in the synthesis and the release of epinephrine, also known as adrenaline. The cortex, however, is important for the synthesis of corticosteroids. We're going to take progesterone and we're going to make two different kinds of corticosteroids, glucocorticosteroid and mineral corticosteroid. Now, each of these has a different role. Glucocorticosteroids, such as cortisol that's shown here, have three main functions. They increase and maintain serum glucose levels by stimulating gluconeogenesis, especially in the liver from amino acids and from fats. They also stimulate fatty acid breakdown, and they stimulate amino acid transport to the liver. They also prevent the uptake of glucose in adipose and in muscle tissue. Glucocorticosteroids also have an important anti-inflammatory role in that they reduce tissue inflammation, and they act as immunosuppressants to reduce the immune response to antigens. In this diagram, we see the mineral corticosteroid aldosterone. Its main function is to increase blood volume and increase blood pressure. This mineral corticosteroid acts on the kidneys to cause retention of sodium in the system rather than its excretion in the urine. And because of this, water follows the sodium because of osmotic pressure, and water is also reabsorbed rather than excreted. Now, this increase of water in the blood will increase blood volume and thus increase blood pressure. The excretion of potassium ions is also affected by aldosterone. Now, androgens include testosterone. The function of this hormone is to promote male secondary sexual characteristics and to affect the libido, energy levels, immune response, and as a protection against osteoporosis. The site of synthesis of testosterone are Leydig cells of the testes in males and follicle cells in the ovaries of females. Testosterone is also produced by the adrenal cortex in both sexes. In this micrograph that's shown here, we see Leydig cells that are found between seminiferous tubules and the testes. These Leydig cells help to support the growing sperm much the same way as follicle cells help to support the growing egg. There are two main types of estrogen, estrone and estradiol. Estrone has a ketone and estradiol has two hydroxyls, and so it is a diol. The function of estrogens is to promote female secondary sexual characteristics and to regulate the menses. The sites of synthesis of estrogens are the follicle cells of the ovary, where they help to maintain and promote the development of the egg, and in the placenta during pregnancy. Estrogens are also produced to a lesser extent in breast tissue, in adrenal glands in both sexes, and in the liver of both sexes. Additionally, in fat cells. This is the end of Lecture 29. Have a great day.